Okie dokie. So, lecture 2 on molecular diagnostics is chromosomal structure and mutations. Again, I put my contact information there. Terms that we kind of already went over and touched on in some of the other lectures were diploid, which means you have 2 complete sets of chromosomes, and in humans, that's 46. Haploid means that you have 1 set of chromosomes, which is 23 chromosomes. The sequence is the order of nucleotides. The phenotype is the trait resulting from transcription and translation, or like your expression, so how you look or what's expressed. Your genotype is the DNA sequence responsible for the phenotype. And a mutation is a DNA sequence change present in a small percent of the population. A polymorphism is a sequence variation present in at least 1 to 2% of the population. And a balanced polymorphism is a genetic alteration with both negative and positive effects. Therefore, it remains in the gene pool. And an example of that is sickle cell. Because you, because sickle-shaped cells, prevent you from getting infected with malaria, it helps. I did have malaria when I was in Nigeria a few years ago. I do not carry the sickle cell trait, so there you go. I got sick. It was a fun time. Don't ever get sick in a third world country when you're not by a hospital. It's a good time. Euploid means the normal number of chromosomes, and aneuploid means an abnormal number of chromosomes. That should mainly be review for all of you because I remember teaching this in high school. So, as far as types of mutations, you can have a gene mutation, a chromosomal mutation, or a genome mutation. So, a gene mutation would affect a single gene. And it's often a small sequence change. So one of the most common ones that has a small sequence change is CF, or cystic fibrosis. So there's lots of tiny mutations that can cause that gene to be ineffective because it's a transport protein. So it doesn't pump your chlorine ions across the cell membrane appropriately. And there's lots of different tiny little mutations that can cause somebody to get cystic fibrosis. We talked about that we used to determine, like, I think we used to detect the 39 most common mutations, but we no longer do cystic fibrosis testing in molecular. A chromosomal mutation is a change in chromosome structure that involves more than just one or a few bases. It's moving thousands or millions of base pairs to a different region or new chromosome entirely. So an example of that is BCR-ABL. And did you guys do that yet? Like learn the cancers and everything? I don't remember when that happens. I think that's at the end. Did you guys learn about BCR-ABL and all of that good stuff?
Not in this program. Not yet.
That'll come. That'll come. You'll learn about it. When do you do hematology? Is that next?
Yeah, second when they get back from their phase 2 of this time.
Okay. You'll learn it soon. Don't you worry. Anyways, that's a rearrangement of chromosomes, causes BCR-ABL. You'll learn about all of that fancy stuff. And then genome, you can have a change in the number of chromosomes, usually an increase because a decrease is often incompatible with life. So Down syndrome, who knows what causes Down syndrome? What's the genome change?
I think it's trisomy 21.
Yes, trisomy 21. Good job, Colby. So you have a whole extra copy of the 21st chromosome. Okay, so we talked about the chromosome morphology the other day. So you go from your DNA double helix, you wrap your DNA around the histones, then you wrap those histones even tighter to get chromatin, and then that chromatin wraps even tighter to make the chromosome. Um, so yeah. You have your P arm, which is the short arm, and your Q arm, which is the long arm. Your telomeres are at each end, and then your centromere um, is not always at the center, but we will talk about that here on the next slide. So there's a few different locations that your centromere can be located. Metacentric means middle. Submetacentric means near the middle, but it can either be slightly higher or slightly lower. Then you have acrocentric, which means towards the end, and then telecentric, which means like at the complete end of your chromosome or at the telomere. We don't have any of these guys in our genome, but we do have quite a few of these little guys and all of those. Um, So visualizing metaphase chromosomes. So metaphase, who can remember what's metaphase? Back to mitosis. What is metaphase? What happens during metaphase?
I think that's when the chromosomes line up in the center.
Yeah, M for middle, right? So most of your chromosomes are right in the center of your cell. The chromosomes are condensed, they're nice and they're gonna arrange, and it's gonna be an amazing time and they're gonna split so that they can duplicate. So what comes after metaphase? Who can tell me this is not anything important, but what comes after metaphase?
Anaphase.
Yes, anaphase, and that was Reagan. Good job, Reagan. They move away, right, from the center. But we want to stop, when we're doing chromosome analysis, we want to stop it in metaphase because all of our chromosomes are really closely located towards the middle of the cell. And then that's where we're going to be able to capture a picture of those and then do an analysis on those pictures. So how we do that, we culture the patient cells. We use our PMI and we use, tissue culture flasks, and we use phytohemagglutinin or PHA to stimulate cell division. That way we're getting lots of cell division. That way we have more cells that we could potentially catch in metaphase when we go to do our analysis. Clogmed arrests cells in metaphase, so we treat them with that. And then a 3-to-1 methanol acetic acid fixes those metaphase chromosomes for staining. So we want to fix them. So I have some pictures here. This is an added slide. And I wanted to show you, this is where we can arrest the cells in metaphase, and depending on the treatment of them, they can be either longer and you can see more bands, or they can be shorter and more densely packed. You don't want the chromosomes too long and you don't want them too densely packed. You want to get a nice banding pattern, and you want to be able to easily differentiate between the multiple different chromosomes. You don't want them too far spread out because then they get really hard to, like, sometimes they overlap and crisscross each other, like in this one. And when we do the analysis, what we end up having to do is we take these slides where we fix these cells on, and we take pictures of these metaphase cells. These are ruptured and we have them in metaphase. We lyse them. We take a picture of it on the slide, and then we take that digital image. And what we do is we zoom in and we crop each of these chromosomes, and then we put them in the karyotype. So we'll drag and drop them into whatever chromosome order that they fit in. And a karyotype is the arrangement of chromosomes from biggest to smallest, and then your sex chromosomes fall in last. So it takes a lot of training and a lot of time in order to identify which chromosome is which, based on the banding pattern. Sometimes this training can take up to a year to get really good at it. And you can't just look at one cell. So when we do a karyotype analysis of these cells in metaphase, a lot of times we're analyzing at least 10 different cells, because sometimes like a random chromosome, even though it's here, it should be in that cell, maybe it floated out of the field of view when you were doing your analysis. So maybe if you just did one karyotype on it, you might be missing a chromosome, or maybe two cells were really close and there you've somehow found an extra chromosome randomly in your picture or your field of view of that chromosome. So you always want to take an average of 10. Sometimes that's hard though, but yeah, we try to do an average of 10 different karyotypes on the same patient in order to get an overall result. So you can see that we have a variation of what these chromosomes can look like depending on when they are stopped in metaphase. So you have very short and dense ones, and sometimes that can be really hard. Some of these look very, very similar, right? And then you get the elongated ones, which this one's a pretty good one, and this one's a pretty good one. This is okay too. You can see some distinct bands. You can tell there's a difference in length, but even some of these get a little crazy. When you get to the small ones, it gets a little hard if they're too compact. Any questions so far?
I don't have a question, but can you hold on a second? Nicole can't get in, and I'm trying to figure out how to add her.
Yeah.
Jen, I just got in.
Oh, you're in? Okay, cool.
Yeah, thank you. Sorry.
You're okay.
You're welcome.
Any other questions before I move on?
Proceed with.
Okay, so how do we visualize these bands? We use a lot of different staining to visualize them. Most of the time, the most common one is the G band. We use a Gamsa stain, but you can do Q band, which is fluorescent. We don't really use that. R bands are the opposite of G bands. So you can see here's our G, and then R's are opposite. So we have this dark region where we had the purple region, and we have purple regions where we had the dark regions. And then C bands are only when you are looking at the centromere. And typically, we do this just as an easy, quick way to identify extra chromosomes or missing chromosomes. So we can just stain all the centromeres and then just count the centromeres that we see, and that's it, right? So it's really nice. And then the number of bands equals resolution. The greater number of bands, the higher the resolution, and the more details you can see. If you go back to this slide, you can see, I don't know if I'm sharing my screen, can I like zoom in? Yes, I can. Did it zoom in for you guys?
Yes, it did.
Okay. So you can see, what if you are spread out and you have maybe a deletion of this little band right here, you might not have been able to catch that on this karyotype up here. So the higher the resolution, or the more bands you can see, the easier it is to be able to detect deletions or duplications or something like that. But again, you don't want them too long. So it's finding that happy median of a good resolution without getting too crazy. So a karyotype, again, it's a direct observation of metaphase chromosomes. And again, we talked about how they're arranged by size, and then your sex chromosomes are at the end. And again, we can detect aneuploidy, translocations, large deletions or insertions, and inversions. Now, again, this takes a lot of training, because your chromosome might seem really long if it has an insertion or a translocation. You really have to be trained to determine what chromosome it is, where the translocation came from, where the break point is, and all of this other stuff. So the people in cytogenetics are bomb at this. Like, they're so good. And again, we go by G-band staining here. So how we report karyotypes, we use the International System for Human Cytogenetics Nomenclature, or ISCN. So we always include the number of chromosomes first, and then the identity of the sex chromosomes, and then any abnormalities. So, right, 46 is a normal number of chromosomes, and XX would be obviously a normal female. 46XY is a normal male. 46XX DEL means deletion. You would then put the chromosome number in the first set of parentheses, and then you would put either a P or Q for short arm or long arm, and then the location. And you would find all of this information. Obviously, there is we use books, right? We use reference materials to tell us, okay, what band is this? What arm is it from, right? So we have chromosomes that, you know, are lined up, and we can tell what chromosome is what based on the banding pattern from textbooks and resources that we have. So that's very, very nice. And then let's say we have number 47, XY. So this has an extra chromosome somewhere. It's a male. And then we identify with a plus or a minus. Obviously, we have a plus of the 21st chromosome. So this person would have Down syndrome because they have an extra 21st chromosome. Does the nomenclature make sense?
Yes, it does.
Yep.
Okay, so this is a normal karyotype. We have 2 copies of everything, right? And then we have an X and a Y. So the karyotype would be 46XY, and that would be a normal male. Can anybody give me the karyotype of this slide? I'm just gonna call on somebody. Nobody volunteers an answer. What about Jake? Jake, what's my karyotype on this slide?
Not gonna be perfect at this one.
I'm not giving you any like chromosomal mutations. Maybe a genome mutation though. So what do you think? What do you think we got going on?
May I phone a friend possibly for help on this?
No, I'll help you. I'll help you. I'll walk through it with you. How many chromosomes are there in there? So you start with the number of chromosomes. How many chromosomes you got?
So you have what looks like to be 22 pairs of chromosomes in there. So, and you have your 2 pairs of sex chromosomes. So it looks like, I think you have, well, in this case, looks like in this case, with X, isn't that just one pair? Isn't that just one chromosome on the X though?
It is only one chromosome on the X, because if it's a female, it'll have 2 X's. If it's a male, it'll have X and a Y.
Oh, that explains it. I was getting a little confused on the. I was a little confused when it showed just the one on the X, and when you said the, can you show me what it is? I was like, oh wait, why is there only one on the X? I got a little confused on
No,
that.
you're okay. So how many chromosomes are there total? Let's start with the total number we have. So you said we had 22 complete sets, so that would be 44. So if you add those sex chromosomes, what's our total number of chromosomes now?
Well, in this case, that would be 40. Well, with the X one, that would be just 47.
47, correct, because you have 44 with our autosomes, and then we have 3 sex chromosomes. So we have an extra one floating around somewhere. So then we list our sex chromosomes. So we have 47, then we would have comma, and then what would we list as our sex chromosomes? Tess, can you answer that?
Would
Would it just be like XY?
Would it be XY or would it be XYY?
Yeah,
Well,
XYY.
XYY.
XYY. Okay, good job. And then in this case, obviously you have an extra Y, but you already listed those sex chromosomes, so you don't need to say plus Y. But yeah, does that make sense? But if it was like plus, then if you had a third 15, you would just go plus 15. But since you're listing those X and Y chromosomes out, you don't need to identify. But yeah, good job, guys. Okay, does anybody want to take a stab at this karyotype?
Would it just be 46XX? Or would it be 44XX?
Okay, it'd be 40. It's not 46 or 44. Look really close, but XX is correct. Anybody else want to chime in? Help a friend? Grace did a really good job.
Wouldn't that be 47 in this case? Because there's an extra found on number 21.
Correct. So you would say 47, comma, XX, comma, and then what would you do?
Would you put plus 21?
Perfect, Tess. You guys are rock stars. I didn't give you anything hard like a deleted on Q, whatever. But yeah, overall, yes. Great job, guys. Okay, so here's some chromosome structural abnormalities. A translocation is where one chromosome and another chromosome, they like switch and they're not supposed to. So this isn't like necessarily like crossing over when you get, genetic rearrangement in meiosis, right? It's when a completely separate number chromosome crosses with another one and they switch, a piece breaks off and switches spots. A deletion is when you delete a region of that gene or the chromosome. You can have an inversion where maybe it breaks apart and flips over and then it goes the other way. See, again, this is why you have to be very, very trained in identifying chromosomes based on bands. Because if you have a little inversion and it's just flipped and you're like, oh my God, this doesn't look right. Like, what is going on? And you're looking at the textbook examples of it, you really have to be trained to identify the break point of it, what chromosome it is, and all of that good stuff. So it takes a long time to get really good at it. An isochromosome is when you have two of the same chromosome, right? Because you have a copy from mom and a copy from dad. And you lose the short, in this case, it loses the short arm and you have two long arms that are connected. And it doesn't show whether they're inverted or not. That doesn't matter. It's just saying like now your two chromosomes are one and you're missing parts, right? It could be that these two short ones also made an isochromosome as well, but it's only showing the big one here. You can have an insertion where one part of a chromosome gets inserted into a separate chromosome. Maybe it got deleted from one and found its way inserted into another, which would essentially be, a translocation. But that's okay. Well, not necessarily a translocation. I lied. You could have a ring chromosome where the telomeres kind of get deleted off, and then the chromosome ends fuse together. And then a derivative chromosome can be where it's essentially newly derived. Maybe there was a translocation. These translocated chromosomes are now the derivative chromosome. So I have a definition. I added the definition to an isochromosome and a derivative chromosome into the notes section of this slide, just because I know that they're not they're not talked about much. So, but a translocated chromosome like this, let's say this is chromosome 2 and this is chromosome 1. This is a derivative chromosome, right? It's a newly resulted chromosome. So how do we detect all these abnormalities besides just looking at karyotypes? We can use FISH. I don't know what happened. I'm so sorry. I think I hung up on you guys. What was the last thing you guys heard?
We can use fish.
Okay, you can use a fish. Perfect. Hold on, I gotta set my screen back up again because I suck at life. I'm so sorry. I rested my elbow on my headphone thing and I hit the hang up button because I'm amazing. Okay, so can you still see my screen or do I need to reshare it?
We can see it.
Okay, perfect. Okay, we can use FISH, which is fluorescent in situ hybridization. And you hybridize, right? We talked about hybridizing in PCR where you hybridize those probes and primers. You can hybridize, they have probes as well in FISH. So you hybridize complementary gene specific or region specific fluorescent probes to chromosomes. So you can look for maybe if you based on the kid's symptoms or the disability that this kid is presenting, you might have a gut feeling that you think that this specific gene is missing, right? You can find a probe and then use FISH to target that gene and see, hey, do you have 2 copies of it and are they on the right chromosomes? So if you do interphase FISH, no culturing is needed. What is interphase? Who can jog my memory of what is interphase when we're talking about cell cycle?
I know it's the start of it.
Okay, so it's not necessarily the start of it, but it's where your cell is most of the time, right? Most of the time your cell is in interphase. It's just chilling. You're not condensing your chromosomes. That's when the cell cycle, like you get into all of that good stuff when you're like lining up in the middle and going away. That's more like the cell division. Interphase is where the cell spends most of its time. So you're not condensed, you're just kind of, you want to be uncoiled, not necessarily densely packed into chromosomes because you're working, you're doing your regular cell processes. You need to be constantly accessing your DNA for transcription and translation to take place. So your DNA is kind of spread out a little bit. It's not nice and packed. That's where you see like just like the nucleus is a blob, but you can still use FISH because those probes will still find those regions of DNA to bind to. So you can still use FISH and interphase and you don't need culturing. Again, I have some YouTube links here. We are going to skip those because you guys didn't want to do that together, but you can do that on your own time. So what are some clinical applications of FISH? So again, identification and characterization of the number and structure of chromosomes and their abnormalities. We can detect microscopically. So we can detect microscopic invisible deletions. So we might not be able to see every single thing that's wrong on those chromosomes in the karyotype, right? We can only detect things that are like, oh, I forget. There's a certain number of base pairs with a karyotype that you can detect, but you got to have a big chunk missing for us to be able to detect it with your eyes. You can detect the use of subtelomeric aberrations. You can do prenatal diagnosis of common aneuploidies. So you can detect things that our eye in a regular karyotype would not be able to detect by using fluorescent probes. It's a great, great time. Oops. Okay, so there are 4 different kinds of FISH probes that are common. And I have pictures of what each of those look like on the next slide, and those pictures were not in the original PowerPoint. So chromosome-specific centromere probes. So we can we can stain all of the centromeres of all of the chromosomes, or you can pick a very specific chromosome centromere stain. It can hybridize to the centromere, and you can detect aneuploidy in interphase and metaphase with this. So let's say you are looking at a centromere stain that detects all centromeres. You could just do a quick count. Do you see 46? Do you see 46 fluorescent dots? If you see 46 fluorescent dots, you're Gucci, right? You know you have all your chromosomes there. If you are only looking for, let's say, sex chromosomes. So let's say you're looking for the centromere on the X chromosome and you're female. You would expect 2 X centromeres to show up. If you're male, you would expect only 1 to show up. If you had trisomy 21 and you were targeting only the 21st chromosome centromere, you would expect 3 dots to show up. So it can detect aneuploidy in interphase or metaphase. Chromosome painting probes. So we have these fancy, beautiful things, and they make your chromosomes all colorful and easy to identify. They can just be very chromosome specific, so the whole chromosome's one single color, but they've come a long way where they do specific gene regions are different colors, and they will bind and paint the chromosomes lots of different colors, and you can detect deletions and insertions based on that as well. But again, these guys have to be in metaphase because in interphase, because your DNA is so spread out, it's just going to be one blob of color, right? It's not going to look pretty. And I'll show you on the next page why. Unique DNA sequence probes. So if you're just targeting one gene of interest, you can take a very specific gene sequence probe and stain it. So you can detect rearrangements, deletions, amplifications. Maybe you have multiple copies of that gene. Okay, so that probe will bind and bind and bind, it'll be a brighter signal. And then you can use telomere specific probes. So it can hybridize to subtelomeric regions, and you can detect telomere deletions or rearrangements. And telomeres are important. The shorter your telomeres are, the more likely you are to get mutations and things. So this is a general centromere probe, right? You can see that each of these dots represents a centromere on a chromosome, and you can count those and say, yeah, I have 46, or oh, I have 49. I don't know. This is a chromosome painting probe. It's come a long way, but you know, this will come with an ID where like the kit says, yes, these are all the colors that chromosome 1 should be in this order. And if they're not in that order, right, and it's not in that color, you can say, oh my gosh, I must have maybe a deletion, or I have an insertion somewhere, or yeah, I don't know. You can have an inversion. Maybe this red and yellow are going to be flip-flopped. So you can more easily detect certain things when you have probes. And then gene-specific probes. Right here, we're going to target this gene. And then you can see, yes, I have 2 copies of it. And then we can actually see it fluoresce on these guys, and bam, bam, they're right there. And then our telomere probes. So you can see that the red dots are the ends of the telomere, and you can see this is like interphase, right? They're not all condensed. It's all spread out, whereas this is more metaphase, right? We have very condensed chromosomes. You can see all the little red ends. And there you go. Any questions so far? Fish probes are beautiful. They're a great time. And you get to use the fluorescent microscope. It's fun. Okay, so again, I wanted to give you some examples of genetic abnormalities by interphase. So again, if you are just looking for a specific chromosome and you just choose a probe that hybridizes to, let's say, chromosome 1, okay, a normal diploid cell, it would have 2 bright spots. If you had an extra copy, you would have 3. If you had a deletion, you would have 1. You don't need to be in metaphase in order for this to happen. Structural abnormalities by fish. This you should be in metaphase for, especially because if you're looking for a structural abnormality on a chromosome, and let's say you want to make sure that let's say these 2 areas should be close together, or maybe they shouldn't. Okay, you have this chromosome where you are stained black and you have this chromosome where you're stained purple. You got 2 copies of each, you're good to go. Well, we had a translocation. So now you can see that this purple and this black probe, right, are really close together, and that kind of gives you an off color, right? It's emitting maybe like a grayish color, and there's only 3 dots instead of 4. So you can say, oh my gosh, these two must be very close together on the same chromosome because, man, they're expressing a different color and I only see 3 dots. So if you had a reciprocal translocation of a product, right? Let's say we had this gene or this probe that, fluoresced in more than one location or spanned a very large location on the chromosome, you can have 2 gray dots at that point, right? This is close. Maybe you can see 2, like a purple and a black dot that you can make out that are really, really close. And then this might be, they might be even closer where they broke and came together. Does that make sense? Yes, no? You can also see break points, so break aparts if they're supposed to be close, right, on a chromosome, and then they break apart. You can also detect a translocation by that way. So there's lots of ways to use these probes to do what you want them to do. It's a great time. So much flexibility. You can also do it by chromosome painting. So we're looking at chromosomes 1, 2, and 4 that are very bright, right? You can see that chromosome 4 here has a little bit of another chromosome on it. This was yellow, and now it has a little red spot. And now we have also a little red spot on this little yellow guy. We've got translocation going on. It's craziness. Crazy. We also do a lot of microarrays. So microarray is like a newer technology, and it'll actually take all of your information and tell you whether you have one copy allele A or allele B for another copy, and then it'll tell you maybe you have A and B. Things to the right of these chromosomes are additions. Things to the left of each chromosome are deletions. That's typically how they're reported. And these are just generic pictures of the chromosomes. So it'll show you, oh, this region on your chromosome was deleted. This whole chromosome is duplicated, right? Because that whole chromosome it has an extra line to the right. This one has just 2 duplications of maybe this telomeric region. Obviously, with somebody with this many issues probably is not viable because that is a lot of genetic issues right there. A lot of additions and deletions. Again, this one shows the same thing, just with a little clearer viewing of the banding, and it actually says like, okay, things to the right are additions, things to the left are deletions. This is actually what a microarray looks like on the computer once you do the analysis. So you have this thing at the top called your log-to ratio. Then you have, do you have the allele A, allele B, or do you have both? Okay, do you have A and B? So some of these spots you have only allele A. Some of these spots you have allele B, and some you have both A and B. You might, because mom and dad each give you a copy. They might be the same copy. They might be different copies, right? You can have a deletion. You can have an addition. So, and then you have a copy number. So your copy number should maintain the same across the whole thing. If we look at this guy, these are looking at 3 different genes. Okay, we have our ratio, and you could see the ratio drops a little bit on the log scale. Something must be happening. Our copy number also drops when we get to the CFAX gene. So chances are we have a deletion. We can also see that we get just like either allele A or allele B. We don't get both A and B being present, which is very, very, very rare. And these are just little, these little dots. They represent lots and lots and lots of different genes. So we have a deletion of a huge region right here. So on this C, again, we have this log-2 ratio, right? We have a decrease in that log-2 ratio. Again, we have a huge deletion of the CFAX gene. We also have a micro deletion here of the CFA37 gene. You can see that there's no A and B at the same time. There's only A or B. And here you can see that there is a duplication. So we have extra lines and our copy number goes up. And so does our log2 value. These are also very hard to read, and it takes a lot of training and a lot of time in order to be proficient at it. So yeah, it's a good time. That's it that I have for microarray results.